It's chapter 34, Nucleic Acid Structure and Function. And let me tell you, this chapter does not mess around.
No, it starts at the atomic level. It really builds the entire concept from the ground up all the way to the complex machinery of life.
It's what you'd call foundational knowledge. If you really want to understand how a virus works, how a cancer drug is designed, or even just how you manage to exist as a coherent being from one moment to the next,
Yeah.
you have to get this stuff.
You really do. The chemistry underpins everything else.
But before we jump into the really heavy chemistry, the hard stuff, I think we should set the stage a little bit because it wasn't always obvious that DNA was the main character in this story.
That's absolutely right. If you go back to the early part of the 20th century, all the smart money was on proteins.
Why proteins? What was the logic there?
Well, just think about it from a complexity standpoint. Proteins are built from 20 different amino acids,
Yeah.
20 different letters in their alphabet,
Okay, so a lot of variety.
a huge amount. And they fold into these unbelievable, intricate, three-dimensional shapes. They're the enzymes, the motors, the structures. They do all the work in the cell.
So they looked at DNA and what? It just seemed too simple.
It seemed boring.
Yeah.
It only has 4 components, just 4 letters in its alphabet. Scientists looked at that and thought, how could something so simple, so repetitive, possibly hold all the information needed to build a human being?
It feels like trying to write, I don't know, the entire Encyclopedia Britannica using only the letters A, G, C, and T. It just doesn't seem mathematically possible.
That was the intuition, exactly. It seemed like it had to be a structural molecule, maybe some kind of scaffold, but the information, that
Yeah.
had to be in the complex proteins.
So what was the turning point? When did the tide start to shift?
The big aha moment, or at least the first major tremor that shook that old idea, came in 1944. It was a series of experiments by Avery, McLeod, and McCarty.
And they weren't even working on, like, human genetics or anything glamorous. They were working with bacteria.
Pneumococcus bacteria, the stuff that causes pneumonia. And the setup was brilliant in its simplicity.
Yeah.
They had 2 strains of this bacteria.
One harmless, one not.
Right. A rough strain that was harmless, and a smooth strain that had this protective capsule and was lethal to mice.
Okay.
And the key discovery from earlier work was that you could mix dead lethal bacteria with live harmless bacteria, and something from the dead ones would transform the live ones.
They would become lethal.
They'd start making that protective capsule and become lethal. So there was some transforming factor being transferred. The question was, what is it?
Is it the proteins? Is it the DNA? Is it the RNA? What's the active ingredient?
Exactly. So what Avery, McLeod, and McCarty did was methodical. They took the extract from the dead lethal bacteria and treated it with different enzymes. First, they used an enzyme that destroys proteins.
Okay, get rid of the prime suspect.
Right. They destroyed all the protein,
Hmm.
and transformation still happened. The harmless bacteria still turned lethal.
Just not protein.
Not protein. Then they used an enzyme that destroys RNA. Did the experiment again.
Right.
Transformation still happened. So it's not RNA either.
Which only leaves one thing?
The last suspect. They used an enzyme, a DNase, that specifically chews up DNA. They added that to the extract. And for the first time, transformation stopped.
The harmless bacteria stayed harmless.
That was it. That was the smoking gun.
Mm.
They proved that DNA wasn't just some boring scaffold. It was the transforming factor. It was the chemical basis of heredity.
And that just shifted the entire world of biology. Suddenly it wasn't about some vague, mystical life force. It was about hard chemistry.
A specific molecule with a specific structure.
And that's our starting point today, the chemistry. So let's dive into that. Let's look at the monomer, the single brick. If we're building this giant Lego castle of life, what does one single Lego brick look like?
Okay, so the fundamental unit is called a nucleotide. Or, to be really precise for DNA, it's a deoxyribonucleotide.
Deoxyribonucleotide.
And you have to visualize it as this distinct 3-part structure. They're all connected.
Okay, let's break it down. What's the central piece? The hub.
At the very center of it all, holding everything together, you have a sugar molecule. And it's a very specific sugar called 2'-deoxyribose.
Now, I want to pause you right there because the text makes a huge deal about that name, specifically the deoxy part.
It's arguably the most important part of the name. Deoxy just means minus an oxygen.
Right. And if you look at the little carbon ring that makes up the sugar, chemists label the carbons 1', 2', 3', and so on.
Yep.
And at that specific spot, the 2' carbon, there's just a hydrogen atom, no oxygen.
Correct. Now, if this were RNA, which we'll get to later, there would be a hydroxyl group there, an OH, an oxygen and a hydrogen.
Okay, so to a layperson, missing one single oxygen atom seems like, I don't know, a typo. It seems totally insignificant.
It does, doesn't it? But chemically,
Mm-hmm.
it is the difference between a building that can stand for 1,000 years and one that crumbles in a week. It's a massive, massive difference in stability.
So walk us through that. Why is having that extra oxygen there so destructive?
It all comes down to a type of chemical reaction called a nucleophilic attack. That hydroxyl group, the OH, hanging off the 2' spot in RNA, is reactive. The oxygen is electronegative, it's aggressive.
It's looking for trouble.
It's looking for trouble, exactly. And in alkaline conditions, or even just slowly over time in water, that oxygen can actually swing around and attack the phosphate backbone of its own chain.
So it's self-sabotage. It literally cuts its own lifeline.
Precisely. It causes hydrolysis.
Yeah.
The chain just snaps. Now, for RNA, that's almost a feature, not a bug. RNA is meant to be a temporary message, a post-it note. You want it to be easy to break down and recycle.
But DNA is the master archive. It's the blueprint in the vault. It needs to last for the entire life of the organism, maybe even longer.
For millennia, in some cases.
Yeah.
So by removing that one reactive oxygen atom,
Evolution basically rust-proofed the molecule.
that's a perfect way to put it. By stripping away that oxygen, the sugar becomes chemically inert at that position. It can't attack the backbone. That single atomic deletion is the main reason we can recover DNA from, say, Neanderthal bones that are 50,000 years old. You could never, ever do that with RNA.
That's just fascinating. So the D in DNA is fundamentally a stability hack.
It's the ultimate stability hack.
Okay. So the sugar is the central hub. Let's talk about the other two parts. What's attached to it?
All right, sticking off what we call the 5' carbon of the sugar, you have the phosphate group. This is a phosphorus atom surrounded by oxygens. And this is crucial because it's the connector. It's the part that's going to form the chain.
It's the glue.
It's the glue, yes. And it also carries a negative charge, which is going to be really important later.
Okay, so phosphate on one side, what's on the other?
On the other side, attached to the 1' carbon, you have the nitrogen space. This is the information. This is the letter in the alphabet.
This is the A, G, C, or T.
Exactly. And as you said, there are 4 flavors of these bases in DNA, but the text classifies them into 2 chemical families, which I always found really helpful for visualizing the geometry.
The purines and the pyrimidines?
Yes, and you should just think of this in terms of size, physical space. The purines are adenine A and guanine G. These are the big guys. They have a double ring structure made of carbon and nitrogen.
2 rings. Got it. And the pyrimidines?
Those are cytosine C and thymine T. They're smaller. They consist of just a single ring.
So we have this fundamental big-small dynamic. 2 big ones, A and G, 2 small ones, C and T.
Precisely. And that size difference is going to be incredibly important in a few minutes when we start pairing them up to make the double helix.
Right, because if you paired 2 big ones, the DNA would physically bulge out.
And if you paired 2 small ones, it would pinch inward. The geometry would be all wrong.
So we'll get to the pairing, but first we have to actually build the chain. We have these individual bricks. How do we string them together?
We build the backbone. And this is formed by what's called a phosphodiester bond. It's a very strong covalent bond.
Phosphodiester. Okay, that sounds complicated.
It's not too bad. It just connects the 5' phosphate of one sugar to the 3' hydroxyl group of the next sugar in the chain. So it goes sugar phosphate, sugar phosphate, sugar phosphate, over and over.
Like a conga line.
That's a great analogy. Everybody's facing the same direction. The left hand of one person grabs the right shoulder of the person in front. And this creates something the text stresses over and over again. Polarity, directionality.
This was a huge point. DNA isn't just a random string of beads. It's a vector. It has a direction.
It absolutely does. We always talk about the 5' end and the 3' end. The 5' end is the start of the chain, where you have that free phosphate group on the 5' carbon.
The front of the conga line.
Exactly. And the 3' end is the end of the chain, where you have a free hydroxyl group on the 3' carbon. And biologically, this is a one-way street. All the machinery in the cell, all the enzymes that read or copy DNA, always move in one direction.
5' to 3'.
Always. The genetic code is always written 5' to 3', and it's always read 5' to 3'.
So if you tried to read it backwards, 3' to 5', it would be complete gobbledygook.
Complete nonsense. It's just like reading a sentence in English. The cat sat on the mat means something.
Mat the onsat cat the.
Means absolutely nothing to the cellular machinery. That directionality is non-negotiable.
Okay, before we leave the backbone and build the second strand, there's one more chemical property the text highlighted that seems really important. The charge.
Yes. Those phosphate groups in the backbone, the PO bonds,
Yeah.
They carry a strong negative charge at physiological pH.
So because DNA is this super long polymer with millions of these phosphates.
It is highly, highly negatively charged. This is why it's called an acid, deoxyribonucleic acid. It's a polyanion.
And that must create some drama, right? I mean, basic physics tells us that, like, charges repel. If you have this long string that's negative and you try to put another long negative string next to it,
Yeah.
they should hate each other.
They do. They repel each other fiercely. That creates a massive electrostatic repulsive force that the structure has to somehow overcome to stay stable. If you just stripped everything else away and put two backbones next to each other in a vacuum, they would fly apart.
Okay, so that's a problem the cell needs to
A
solve.
huge problem. And we'll see how it does it, using both the bases inside and ions from the outside when we talk about stability. But just keep that in mind. DNA is a molecule that is constantly fighting its own internal electrostatic repulsion.
Okay, so we've built a single strand. We have a sugar phosphate backbone with a 5-to-3 directionality and the bases sticking off of it. But life, for the most part, isn't single-stranded. We need to talk about the icon, the double helix.
The Watson-Crick model, proposed in 1953, based on, and we have to mention this, based on the incredible X-ray diffraction data from Rosalind Franklin and Maurice Wilkins.
Her photograph 51, which was the key piece of evidence.
It was the key. It showed the helical shape, the dimensions. It was the Rosetta Stone for the structure.
So visualize it for us. We have the two strands we just built. How do they come together?
First, and this is a concept that trips people up all the time, they're antiparallel.
Antiparallel.
It means one strand runs 5' to 3' going down. The other strand runs 5' to 3' going up. They're oriented in opposite directions, upside down relative to each other.
So it's like a two-way highway, the northbound lane and the southbound lane.
That's a perfect analogy.
Yeah.
And they wind around a central axis, and it's a right-handed helix. So if you were to look down the barrel of it, it twists clockwise as it moves away from you.
Okay, so they're twisting. Where are the parts? Where are the backbones and the bases?
The sugar phosphate backbones form the outside rails of the ladder. They're on the exterior, exposed to the water in the cell. And the bases, those A's, G's, C's, and T's, they point inward.
They form the rungs of the ladder?
They form the rungs, exactly. They're tucked inside, perpendicular to the central axis, facing each other.
And this is where the pairing rules come in. This is Erwin Chargaff's great discovery. The pairing isn't random.
No, not at all. He found that in any organism's DNA, the amount of A was always equal to the amount of T, and the amount of G was always equal to the amount of C,
Chargaff's rules.
which led Watson and Crick to the pairing model. A always pairs with T, and G always pairs with C.
And let's connect that back to the size thing we mentioned. A and G are the big purines. T and C are the small pyrimidines.
So this means a big purine is always pairing with a small pyrimidine.
2 rings plus 1 ring.
Always equals 3 rings wide. This keeps the diameter of the helix perfectly uniform all the way down. It's exactly 2 nanometers, or 20 angstroms, wide. It doesn't bulge, it doesn't pinch. It's geometrically perfect.
But the pairing isn't just about fitting the space. It's about the chemistry. It's about the glue holding those rungs together, the hydrogen bonds.
This is the key to the specificity of the code. The shapes and the charge distributions on the bases are such that adenine and thymine can form 2 stable hydrogen bonds between them.
Okay, A and T, 2 bonds.
Guanine and cytosine, on the other hand, can form 3 hydrogen bonds.
3. Okay, let's unpack that. That seems like a really important detail. G and C have 3 bonds. A and T only have 2. That implies that the GC connection is stronger.
Significantly stronger. A GC base pair is about 50% stronger than an AT base pair. Think of it like Velcro or a zipper. The more teeth you have engaged, the harder it is to pull apart.
So a stretch of DNA that's rich in GC pairs is physically tougher to separate than a stretch that's rich in AT pairs?
Much tougher. It takes more energy, specifically more heat energy, to break those 3 bonds compared to the 2.
And I'm guessing that's not just a lab curiosity. That has to matter in nature.
It matters immensely. If you look at the genomes of thermophilic bacteria, these are organisms that live in boiling hot springs or deep-sea thermal vents. Their DNA is often highly enriched with GC pairs.
So their DNA doesn't literally melt in the heat?
Exactly. Physics suggests they evolved that way as a thermal adaptation. It's a way to keep their genetic archive from falling apart at high temperatures.
We're going to get to melting in a minute, but I want to stick with the forces holding this thing together. We have the hydrogen bonds holding the rungs of the ladder together horizontally. But the text made a really interesting point. It said the hydrogen bonds actually aren't strong enough on their own to keep the helix stable in water.
You caught that. That's a very sophisticated point that often gets glossed over. We tell students about the hydrogen bonds because they explain the AT-GC specificity. But the real unsung hero of DNA stability is a force called base stacking.
Base stacking. Okay, what is that?
So imagine the bases again. They're flat, planar molecules, like dinner plates or coins. In the helix, they are stacked horizontally on top of each other, one after another, down the core of the helix.
Like a stack of
A
pancakes.
stack of pancakes is a perfect image. Now, inside the cell, there is water everywhere. But these bases, with all their ring structures, are hydrophobic. They hate water.
So they want to hide from it.
They want to huddle together in the middle of the helix, away from the water. And by stacking so tightly on top of each other, they exclude the water. This creates favorable hydrophobic interactions. And there are also these very weak short-range attractions called van der Waals forces that happen when atoms get very close.
And because the entire stack is so long and so tight.
Those tiny forces add up. They add up to a massive amount of stabilizing energy. Some calculations suggest base stacking provides more stability to the overall helix than the hydrogen bonds do.
So it's really a dual locking mechanism. The horizontal hydrogen bonds give you the coding specificity.
A pairs with T, G pairs with C.
And the vertical base stacking gives you the overall structural integrity. It's what keeps the whole thing rigid and stable.
That's it. It's a brilliant piece of chemical architecture.
Now, I want to spend a minute on the overall geometry, because that spiral staircase image we all have in our heads is actually a bit misleading, or at least it's too symmetrical.
It is. The way the sugar-phosphate backbones connect to the bases isn't perfectly symmetrical. The N-glycosidic bonds that attach the base to the sugar are at an angle.
So what does that do to the overall shape?
Well, if you build a spiral staircase where the steps were attached to the central pillar at a weird angle, you'd find that one side of the staircase is wide and open, and the other side is cramped and narrow.
The major and minor grooves.
Exactly. The DNA double helix has a wide, deep major groove and a narrow, shallow minor groove that spiral along the outside of the molecule.
And the text argues that this lopsidedness isn't a design flaw, it's a critical feature. It's the user interface of the DNA.
That is a great way to put it. Think about the problem the cell has. The genetic information, the sequence of A's, G's, C's, and T's, is buried on the inside of the helix.
Right, the rungs of a ladder are in the middle.
So if a protein, let's say a regulatory factor that needs to turn a specific gene on or off, wants to read the sequence, how does it do it? How does it find its specific binding site? Maybe a sequence like GATTC.
It can't just unzip the entire genome to look for one gene. That would be chaotic and energetically impossible.
Right. So the major groove is the solution. It's wide enough to expose the edges of those base pairs to the outside world. And here is the really cool part. The chemical signature of an AT pair looks different from the outside than a GC pair.
What do you mean it looks different?
I mean, the pattern of hydrogen bond donors, hydrogen bond acceptors, and nonpolar methyl groups that are pointing out into that groove is unique for each of the 4 possible base pairings. An AT pair presents a different chemical face to the world than a GC pair, or a TA pair, or a CG pair.
So the protein doesn't need to open the book to read the words. It can just run its fingers along the spine and feel the letters.
It is literally molecular braille. A protein can slide along the DNA in the major groove, and without ever unwinding the helix, it can read the sequence of the base pairs inside.
That is incredible. It completely changes the way I visualize it. It's not a closed vault. It's an accessible, readable surface.
And the major groove is the main landing pad, the primary information highway, for the vast majority of proteins that need to interact with DNA in a sequence-specific way.
Okay, so we've built the structure. It's stable, it's double-stranded, anti-parallel, right-handed. It has grooves. Now, let's mess it up. Let's talk about DNA dynamics, because this thing isn't a static statue. It has to move. It has to open.
It breathes, it bends. And yes, it can be taken apart completely. And that brings us to the process of denaturation, which is just the scientific term for melting the DNA.
When you say melting, you don't mean it turns into a liquid goo like melting plastic. You mean the 2 strands separate.
Exactly. The covalent bonds of the backbone stay intact, but the weaker bonds holding the 2 strands together break. If you take a solution of double-stranded DNA and you start to heat it up, you're adding kinetic energy, the molecules start vibrating more and more violently.
And eventually, that energy is enough to overcome the forces we just talked about.
Right. It overcomes the hydrogen bonds between the bases and disrupts the hydrophobic base stacking interactions. And at a certain point, the 2 strands just unzip and float away from each other as 2 separate single strands.
And there's a specific temperature where this happens, which the text calls the T.
the melting temperature, or TIM, and it's defined technically as the temperature at which 50% of the helical structure is lost. So it's the midpoint of the transition from double-stranded to single-stranded.
And going back to our earlier point about the GC Velcro, this TIM isn't a fixed number. It changes based on the DNA sequence itself.
Absolutely. If your DNA molecule is full of those triple-bonded GC pairs, it's going to have a high TIM. You need to put in more heat, get to a higher temperature to break all those strong bonds.
And if it's got a lot of the weaker double-bonded AT pairs?
The TIM will be lower. It's much easier to melt. You can actually estimate the TIM of a short piece of DNA just by counting its G's and C's.
But the text also mentioned another critical factor, salt. Salt concentration. This feels like a detail we should unpack because it relates back to that electrostatic repulsion we talked about with the backbone.
It's directly related. It's the other side of the stability coin. So remember, the two backbones are highly negatively charged, and they want to push each other apart.
The repelling magnets.
Right. Now, what happens when you dissolve salt, like sodium chloride, NaCl, in water?
It breaks apart into ions. Positively charged sodium ions, Na plus Me, and negatively charged chloride ions, then E.
Exactly. And those positive sodium ions are very attracted to the negative phosphate groups on the DNA backbone. They swarm around the DNA and form a kind of cloud of positive charge.
They mask the negative charge, they neutralize it.
They act as a shield. They neutralize that repulsive force between the two strands. So in a high salt solution, the strands are much happier staying together. The repulsion that was trying to push them apart is gone.
So high salt stabilizes the DNA and makes the TIM go up.
Correct. And if you do the opposite, if you put DNA in very pure water with very low salt,
the shield is gone, the negative charges on the backbones are fully exposed, they repel each other violently, and the DNA melts at a much lower temperature. It's much less stable. That seems like such a in-the-weeds detail, but the text makes it clear that scientists use this property in the lab constantly.
every single day. In techniques like PCR, we're carefully controlling both the temperature and the salt concentration to get DNA strands to separate and come together exactly when we want them to. And we can track this process using a property called hyperchromicity.
Which sounds like a comic book superpower.
It does, but it just refers to how DNA absorbs light. The aromatic rings in the bases are very good at absorbing ultraviolet light, specifically at a wavelength of 260 nanometers.
Okay.
But when they're all stacked up neatly inside the double helix, they kind of hide from the light a bit. They shield each other. This is called the hypochromic effect. The stacked DNA absorbs less light than you'd expect.
But when you heat it up and the DNA melts...
The strands separate, the bases unstack, and they become fully exposed to the solution. And suddenly, they absorb more UV light. The absorbance at 260 nanometers shoots up by about 40%.
So you can literally put a sample of DNA in a machine, shine a UV light through it, slowly raise the temperature, and you can watch the exact moment the strands separate because the solution suddenly becomes less transparent to UV light.
Precisely. You get this beautiful S-shaped curve, and the midpoint of that curve is your tin. It's a very elegant way to monitor the physical state of the molecule in real time.
Okay, so we can melt it. What if we cool it back down? Can we put the toothpaste back in the tube?
Yes, absolutely. This is called renaturation, or more commonly, hybridization. If you take that solution of single-stranded DNA and you cool it down slowly, the strands will start randomly bumping into each other.
And eventually, a strand will bump into its correct partner.
Right. A short sequence will find its complement, an A finding a T, a G finding a C, and form a few hydrogen bonds. This creates a little nucleation site, and from there, it's like a zipper. The rest of the strands rapidly zip back up into a perfect double helix.
And this isn't just a party trick. The text says this is the fundamental basis for, well, for almost all modern genetic diagnosis and technology.
It is the absolute foundation of modern molecular biology. Think about a southern blot, where you're looking for a specific gene in a complex sample. You make a single-stranded probe that matches that gene, you label it, and you add it to your melted sample.
And it will hybridize only to its perfect match.
Exactly. It finds the needle in the haystack. The same principle applies to northern blots for detecting RNA, and even to PCR tests for viruses like the ones used for COVID. They all rely on this incredible ability of a single strand of nucleic acid to search through a complex mixture and find its one true complementary partner.
Okay, we have to touch on one more physics concept before we switch over to RNA. Dicology. The chapter calls it the twisted phone cord problem.
Ah, yes. This is a fascinating area. Imagine you have a long, long rubber band. You hold the ends, and you just keep twisting it.
Eventually, it starts to coil up on itself. It forms these bigger loops and knots.
That's supercoiling. And DNA does the same thing. You have to remember, the DNA in a single human cell is about 2 meters long, and it has to be packed into a nucleus that's about 10 microns wide.
That's like packing 20 miles of thread into a basketball. The packing problem is immense.
It's immense. And to
Yeah.
do that, and also to manage the tension that builds up, when you try to unwind a small section for reading or copying, the DNA gets supercoiled. It gets twisted upon itself.
I'm picturing that old landline phone cord analogy, and it's stressing me out. If you have this tightly wound, super coiled structure, and then you try to pull two strands apart to replicate it, physics says the tension on the rest of the molecule must become insane. It should get wound up tighter and tighter until it snaps.
The torsional stress would be catastrophic. If the cell didn't have a way to solve this problem, the DNA would shatter the moment it tried to replicate.
So the text introduces the solution, a class of enzymes called topoisomerases. But I have to say, the mechanism described here sounds incredibly risky.
It is incredibly risky. It's high-stakes molecular surgery.
You're telling me that to save the DNA from snapping under tension, these enzymes break it on purpose.
That's exactly what they do. A type I topoisomerase acts like a swivel. It nicks one strand of the DNA, just cuts one backbone. This allows the other strand to spin around it freely, releasing the tension. And then, and this is the crucial part, it seamlessly reseals the cut.
It's a controlled demolition and reconstruction.
Precisely. And then you have the type 2 topoisomerases, which are even wilder. They have to deal with two double helices that are tangled up. So they grab one helix and they cut both strands, a double strand break.
That sounds lethal.
It would be, but it's transient. It literally opens a gate in the first DNA molecule, passes the second DNA molecule straight through the gate, and then closes the gate behind it by resealing the break. It's how the cell untangles its chromosomes.
That sounds like actual magic. How fast is this happening inside our cells?
Constantly. Thousands upon thousands of these cut and paste events are happening every second, especially when a cell is dividing.
And you mentioned the risk. What happens if that enzyme makes the cut and then fails to reseal it?
You've just given the cell a death sentence. You've created a permanent double-strand break, which can lead to mutations, chromosome rearrangements, and usually cell death. And in fact, that's exactly how some of our most powerful chemotherapy drugs work.
Really? How?
Yes. Drugs like etoposide or doxorubicin. They don't attack the DNA directly. They're what we call topoisomerase poisons. They gum up the enzyme. They allow the topoisomerase to make its cut in the DNA of a rapidly dividing cancer cell, but they physically block it from resealing the break.
So they turn the cell's own safety valve into a suicide weapon.
Exactly. You're forcing the cancer cell to shred its own genome. It's a brilliant therapeutic strategy.
That is just, wow. We went from bond angles and grooves all the way to a cancer therapy mechanism in just a few steps.
That's the power of understanding the fundamental structure. All the biology flows from it.
All right, let's pivot. We've spent a lot of time on the star of the show, DNA. But the chapter spends a huge amount of its ink on its less famous but maybe more versatile cousin, RNA.
The chemical cousin. Very similar in many ways, but with a few critical differences that give it a completely different personality and job description.
Okay, let's play spot the difference. We've already covered number one, the sugar. RNA uses ribose instead of deoxyribose.
Right, and as we established, that extra hydroxyl group on the 2' carbon makes RNA chemically unstable. It's prone to hydrolysis, especially in alkali.
Which explains why DNA is the archive in the climate-controlled vault, and RNA is like a Post-it note stuck to the fridge. You don't want your permanent records written on paper that dissolves in the rain.
Exactly. RNA is designed to be transient. It's made, it's used for a short period of time, and then it's degraded and its components are recycled.
Okay, difference number two, the bases.
RNA doesn't use thymine, the T. Instead, it uses a very similar base called uracil, or U. So in RNA, the alphabet is A, G, C, and U.
Why the switch? Is uracil better somehow? Or worse?
Chemically, uracil is very, very similar to thymine. Thymine is basically just uracil with a little methyl group, a CH3 tacked onto it. But here's the deep evolutionary insight that the text explains. Cytosine, the C, is chemically a bit unstable. Over time, it can spontaneously denaminate, lose an amino group.
Okay.
And when cytosine denaminates, it turns into uracil.
Oh, that sounds like a big problem.
It is a massive problem for a genetic archive. Imagine if DNA used uracil as one of its normal letters, and then a cytosine somewhere in the code spontaneously broke down and became a uracil. The cell's repair machinery would look at that uracil and have a question.
Is this uracil supposed to be here? Or was this a cytosine that broke?
It would have no way of knowing. It would lose the ability to proofread and correct that specific, very common type of damage.
It's ambiguity.
And ambiguity is death for a high-fidelity code. So by using thymine, which has that extra methyl group tag DNA, creates an unambiguous system. If the cell's repair crew ever sees a uracil in DNA, it knows immediately, that does not belong here, that's a damaged cytosine. Kick it out and replace it with a C.
So thymine is like a verified or official version of uracil specifically for long-term storage.
That's a perfect description. And since RNA is short-lived, it doesn't need that level of long-term error correction, so it just uses the metabolically cheaper uracil.
That's brilliant. Okay, third big difference, strandedness.
DNA, as we've discussed, is a double helix. RNA is typically single-stranded.
And this is a huge but that the text emphasizes. Just because it's a single strand doesn't mean it's a floppy straight line like a piece of spaghetti.
No, not at all. In fact, because RNA is single-stranded, it's free to do something DNA can't. It can fold back on itself.
How does that work?
Regions of the single strand that are complementary to each other can find each other in space and form short stretches of double helix. A pairs with U, G pairs with C. This creates complex three-dimensional origami shapes.
You get things like hairpins and loops and stems.
Exactly. And that intricate 3D structure is what allows RNA to have function beyond just carrying information. Its shape allows it to act like an enzyme, to catalyze chemical reactions. We call those RNA enzymes ribozymes.
So RNA is way more versatile than I gave it credit for. It's not just a simple photocopy of the DNA message.
It's a functional tool. It can be a messenger, a structural scaffold, a catalyst, a regulator. It wears many, many hats in the cell.
So let's break down some of those hats. The text describes several different lives of RNA. There isn't just one type.
No, there are several distinct classes, each with a specific job. The most famous one, of course, is messenger RNA, or mRNA.
The celebrity. Especially since the COVID vaccines made it a household name. This is the copy of a single gene that gets sent out to the protein factory.
Right. It carries the message from the DNA in the nucleus out to the ribosomes in the cytoplasm. But one thing the text highlights is that the population of mRNA in a cell is very heterogeneous.
Meaning it varies a lot.
It varies wildly in size and in sequence, because proteins vary wildly in size. If you need to make a small protein hormone, you'll have a short little mRNA. If you need to make a massive muscle protein like titin, you have a gigantic mRNA.
Now, the text mentions that before the mRNA leaves the nucleus, it gets a couple of accessories, a cap and a tail. It sounds like it's getting dressed up to go outside.
It kind of is. It's a processing step to make it stable and recognizable. At the 5' end, the starting end, a special nucleotide is added called a 5' cap. It's a modified guanosine, a 7-methylguanosine that's added on backwards.
Backwards? What does that mean?
It's a 5' to 5' linkage, which is very unusual. All the other linkages are 5' to 3'. This backward cap acts like a helmet. It protects the start of the message from being chewed up by enzymes that like to nibble from the 5' end.
And it also helps the ribosome recognize where to start translating.
Exactly. It's the start here signal.
And the tail.
That's the polytail, added to the 3' end. It's just a long string of adenine nucleotides, sometimes hundreds of them. A. This tail stabilizes the molecule and also acts as a kind of timer.
A timer?
Yeah. Over time, in the cytoplasm, enzymes start chewing away at that tail, making it shorter and shorter. Once the tail gets too short, the cell knows that this mRNA is old, and it gets targeted for destruction.
So it's a built-in expiration date, a message that self-destructs after a certain amount of time.
Exactly. It ensures that the cell isn't endlessly making a protein it no longer needs.
So that's mRNA, the message. What about the factory floor itself? The text talks about ribosomal RNA.
rRNA. This is the workhorse. It is by far the most abundant type of RNA in the cell. If you were to take all the RNA out of a cell and weigh it, something like 80% of that mass would be RNA.
Because it makes up the ribosome itself.
The ribosome is a gigantic molecular machine, a complex of RNA and proteins. And the RNA isn't just a scaffold, it's the catalytic core. It's actually an rRNA molecule that forms the peptide bond between amino acids. It's a ribozyme.
Now, the text uses these S units to describe the ribosome, the ADS ribosome in eukaryotes.
Right.
And I've always been confused by this. It says the ribosome is made of a large 60S subunit and a small 40S subunit. But 60 plus 40 is 100, not 80. What's going on there?
It's the bane of every introductory biology student's existence. The S stands for Svedberg unit. And it's not a measure of mass or weight, it's a measure of sedimentation rate. Basically, how fast does something sink to the bottom of a tube when you spin it in a high-speed centrifuge?
Okay, so it's about speed, not weight.
Right. And how fast something sinks depends on both its mass and its shape, its drag through the liquid. So when you combine the 60s and 40s parts, the total mass is 100 arbitrary units, yes. But the shape of the combined particle is different. The drag changes. So it doesn't sediment at a rate of 100. It sediments like an 80s particle.
Mystery solved. It's basically hydrodynamics.
Centrifuge dynamics, but yes, exactly.
Okay, third major type of RNA, transfer RNA, tRNA. The text calls it the adapter.
This is my favorite molecule. I think it's the cleverest thing evolution ever came up with. It is the physical Rosetta Stone.
Yeah.
It's the molecule that translates the language of nucleic acids, the sequence of bases, into the language of proteins, the sequence of amino acids.
And it has a very distinct, famous shape. The text shows it flattened out, looking like a clover leaf.
In 2D, it looks like a clover leaf with these different arms and loops. In 3D, it actually folds up into a compact L shape. But the key functional parts are at the two ends of the L.
What's on those ends?
At one end, the 3' end, the sequence always ends in CCA. This is the acceptor arm. This is where the specific amino acid gets attached by an enzyme. So you have a tRNA for alanine, a tRNA for glycine, and so on.
So it's like a little molecular backpack carrying one specific amino acid.
That's a great image. And at the other end of the molecule, in a region called the anticodon loop, there are 3 specific bases, the anticodon.
And this is the part that reads the mRNA.
This is the part that reads the mRNA. If the codon on the mRNA says, for example, AUG, then the tRNA with the complementary anticodon UAC will bind there. And that specific tRNA just happens to be carrying the amino acid methionine.
It's a purely mechanical matching system. It physically brings the correct brick to the construction site based on the blueprint.
Pure mechanics. It is the physical embodiment of the genetic code.
Okay, finally, the text mentions a whole group of RNAs called small RNAs. These don't seem to be involved in building proteins directly. They seem to be regulating things.
They're the regulators, the managers. And it's a field that has just exploded in the last couple of decades. There's snRNA, or small nuclear RNA, which is part of the machinery that splices and edits the mRNA before it leaves the nucleus.
The quality control team.
Exactly. But the really hot topic, the one with huge medical implications, is miRNA, microRNA, and siRNA, small interfering RNA.
Interfering? That sounds aggressive.
It is. These are tiny little pieces of RNA, maybe 20 to 25 nucleotides long. And their job is to hunt down specific mRNAs in the cytoplasm and silence them.
Silence them how?
They bind to a complementary sequence on a target mRNA. And this recruits a protein complex that either chops the mRNA to pieces, destroying it, or just physically blocks the ribosome from translating it.
So it's a way for the cell to say, I know I transcribed this gene and made this message, but you know what? I've changed my mind. Destroy the message before it becomes a protein.
It's a powerful layer of post-transcriptional gene regulation. We call it gene silencing, or RNA interference. It's a way to fine-tune the amount of protein that gets made from any given gene.
And the text hints that this is a huge area for future medicines.
Absolutely. Think about it. If a disease like Huntington's disease is caused by a single bad protein, we could theoretically design a synthetic siRNA drug, a small piece of RNA that we inject into the patient.
And it would hunt down only the mRNA for that one bad protein.
And destroy it. You could silence that specific gene, lowering the levels of the toxic protein without ever touching the patient's actual DNA. It's a revolutionary concept for drug development.
Incredible. It's like software debugging for the human body.
It really is.
Okay, let's move to part 6, the enzymes, the toolbox. We've talked about the structures. Now let's talk about the tools that cut and paste them.
The chapter calls them nucleases. These are any enzymes that degrade nucleic acids by breaking the phosphodiester bonds. And we classify them by how they eat.
How they eat.
Yeah, that's how I think of it. You have exonucleases. These are like Pac-Man. They start at one end of a DNA or RNA strand, either the 5' or 3' end, and they chew off one nucleotide at a time. They nibble from the outside in.
Okay, so that's for trimming the ends. What's the other kind?
Endonucleases. These are the karate choppers. They don't care about the ends. They bind and cut somewhere in the middle of the chain.
And there's a very special class of endonucleases that completely revolutionized science in the 1970s. The restriction enzymes.
These were discovered in bacteria. They're part of the bacterial immune system, a way for bacteria to chop up the DNA of invading viruses.
A defense mechanism.
Right. But what makes them so special for us is their incredible specificity. They don't just cut DNA anywhere. Each restriction enzyme recognizes and cuts at one very specific sequence of bases. And that sequence is usually a palindrome.
A palindrome, like race car or madam, I'm Adam.
Exactly. A DNA sequence that reads the same forwards on one strand as it does backwards on the complementary strand. The famous enzyme E-cor-I, for example, looks for the sequence GATTC. It will scan along a DNA molecule, and it will only cut at that precise spot.
Why was that discovery so important for scientists?
He gave us molecular scissors. Before restriction enzymes, a chromosome was just this gigantic, unmanageable string of DNA. There was no way to isolate a specific piece. But suddenly, with these enzymes, we could cut a huge genome into a defined, reproducible set of smaller fragments.
You could make a map?
You could make a map. And even more importantly, you could cut a gene out of one piece of DNA and paste it into another piece of DNA using an enzyme called a ligase. This was the birth of molecular cloning and genetic engineering, making human insulin in bacteria that all started with restriction enzymes.
And the text mentions the modern successor to this kind of technology, CRISPR-Cas.
CRISPR is like a restriction enzyme on steroids. It's a programmable nuclease. Instead of being stuck with an enzyme that only cuts GATTC, the CRISPR-Cas9 system uses a small guide RNA.
And you can design that guide RNA to match any DNA sequence you want.
Any sequence in the entire genome. So you can tell it, go to this specific spot on chromosome 7 and cut. It's the ultimate tool for genome editing.
It allows us to edit the book of life, not just read it and copy it.
That's right.
All right, we're in the home stretch. Part 7, a quick overview of synthesis, the central dogma. The text outlines the basic mechanics of how we get from DNA to more DNA, which is replication, and from DNA to RNA, which is transcription.
Let's start with replication. The absolute key word here, the core concept, is semi-conservative.
Semi-conservative, what does that mean?
It means when a cell divides and needs to copy its DNA, you don't just make a brand new double helix from scratch next to the old one. Instead, you unzip the original parent helix.
And each of those two old strands serves as a template.
Exactly. Each old strand is used as a template to build a new complementary partner strand. So the result is two new double helices, and in each one strand is old from the parent, and one strand is brand new.
Half old, half new, semi-conservative.
Right. And this is a brilliant system for ensuring fidelity. You are directly tracing the master copy every single time, which minimizes errors.
Okay, that's replication. Now, transcription. DNA to RNA. This is where I have to admit I always get tripped up on the terminology. Template strand versus coding strand.
You are not alone. It's notoriously confusing nomenclature. Let's visualize it very carefully. You have your double-stranded DNA gene, the enzyme that makes RNA polymerase comes along. It has to read one of those two strands to know what RNA to make.
Okay, it can't read both.
You can't read both. It binds to and reads one specific strand. The strand that it physically reads is called the template strand. Sometimes it's called the antisense strand.
Okay, so the RNA molecule that gets made is complementary to that template.
Right. If the template DNA has an A, the new RNA gets a U. If the template has a G, the RNA gets a C. It's standard base pairing.
But then there's the other strand of DNA, the one that was just sitting there, not being read.
That is the coding strand, or the sense strand. And here is the trick, the reason for its name. Because it is also complementary to the template strand.
It has the exact same sequence as the new RNA molecule.
Exactly the same, with the one exception that the DNA has T's where the RNA has us.
So when we look up a gene sequence in a database or see it written in a textbook, we are usually looking at the sequence of the coding strand because that sequence shows you what the final mRNA product is going to look like.
Yes. Even though the enzyme physically read the other strand to make it, it's a convention of nomenclature, but it is absolutely vital for reading genetic diagrams. If you mix up which strand is which, you'll predict the wrong RNA and the wrong protein.
That is a crucial distinction. Okay, we have done it. We've unpacked the monomer, built the helix, explored its dynamics, visited the RNA zoo, opened the enzyme toolbox, and reviewed the basics of synthesis. That is a lot of chemistry.
It is. But I hope the listener can see that it's not just a collection of random facts, it's a coherent system. The structure dictates the function at every single level. The 2'-deoxy sugar leads directly to long-term stability. The 3 hydrogen bonds in a GC pair lead directly to a higher melting point. It all connects.
So, as we wrap up this very deep dive, let's bring it back to relevance. Why does understanding the precise position of a hydroxyl group or the nature of the major groove matter to the person listening right now?
It matters because this is the literal operating system of your body. When you get a viral infection, That virus is a master of hijacking these exact mechanisms. When we diagnose a genetic disease or use a PCR test, we are manipulating melting temperatures and hybridization in a tube.
And the drugs of the future that we talked about, the mRNA vaccines, the siRNA gene silencing therapies, CRISPR-based cures, they are all designed based on a deep understanding and ability to manipulate these fundamental structural rules.
Exactly. We are moving from an era of just observing biology to an era of engineering it. And you cannot engineer what you do not understand at the most granular chemical level.
Now, as we always do, I want to leave our listeners with one final provocative thought to chew on. We talked about the known roles of RNA, the messenger, the transfer, the ribosomal factory floor. But the text very briefly mentioned things like circular RNAs and long non-coding RNAs.
LNCRNAs, yes. A huge and mysterious class.
For a very long time, we called the vast portions of the genome that didn't code for protein junk DNA. It was the dark matter of the genome.
We did. It was an act of scientific hubris, really. We didn't know what it did, so we arrogantly assumed it did nothing. We know better now.
So my question is this. If all the structural rules we've discussed today, the base pairing, the chemical stability, the ability to fold into complex shapes, if all those rules apply to the RNAs that are transcribed from these vast, unexplored, so-called junk regions, what are they doing?
That is the billion-dollar question in biology right now. We're finding that they're not junk at all. They're forming complex structural scaffolds inside the nucleus. They're acting as decoys for microRNAs. They're guiding proteins to specific locations on chromosomes. They might be the software that organizes and runs the hardware of the protein coding genes.
So we might have only just finished reading the table of contents of the Book of Life, and the real complex story is hidden in all the pages we used to call junk.
I would bet my career on it. The more we look at the structure and architecture of the non-coding genome, the more incredible function we find.
And that is where we will leave it. The next time you see that iconic double helix, don't just see a static ladder from a textbook. Try to visualize it as a dynamic, breathing, twisting chemical machine.
It's the most beautiful piece of architecture in the known universe.
Thanks for diving deep with us.
It was a pleasure.
We'll see you next time.